Kathleen said, I am really passionate about. So as Kathleen mentioned, I'm mainly an evolutionary biologist, and during my PhD, I specialized a lot on phylogenetic models. So I basically like to do a lot of computational studies to try to understand the biology of plants. And I happen to love these little cute plants called liverworts. So in today's talk, I want to start by sharing with you why I really like these plants and why I think we need to conduct more evolutionary studies on liverworts. Then I want to talk about two different projects that I worked on during my PhD that span like different organization levels of liverworts, from population genetics to macroevolutionary studies. And then I want to share with you some future research that I'm starting to develop and that I'm super excited about and that should keep me busy for the next few years. So on to the first part, the Oda to the liverworts. Given the fact that this is a talk in the California Botanical Society, I think I don't need to make a lot the argument that plants are amazing, but just in case, let's think about how plants are food. Plants are materials. Plants have so many interesting interactions. Plants are culture and they're medicine. So plants are really at the center of human experience. And from an evolutionary perspective, photosynthetic organisms in general, so obviously including plants, have really shaped the evolution of life on Earth. I really like to think about deep time. So, for example, from the very beginning of life, cyanobacteria changed the atmosphere composition and therefore changed the whole trajectory of life on Earth. Later on, when photosynthetic organisms moved into terrestrial ecosystems, they played a very important role in shaping the soils that then allowed other groups of plants to evolve, some of which took it onto the skies and really created this third dimension, the vertical dimension in which diversity has evolved, and that we know is like really fundamental in shaping terrestrial ecosystems nowadays. But when we think about plants, we very often only think about angiosperms. And there are good reasons for that, because angiosperms are super important, they're super diverse. We use them all the time. But if we think about the whole diversity of plants, here we have a phylogeny in which we have all the plants as you can see here, and we put it in a time scale. Angiosperms account only for this group. And also very diverse now, if we think about the whole history of terrestrial ecosystems, for more than half of the history of terrestrial ecosystems, there were no angiosperms. Yet, most of our understanding of plant biology and plant evolution is based on vascular plants, and particularly angiosperms, perhaps very unbalanced to what we know about other groups like bryophytes that have been around for much longer. And in particular, as I mentioned, I am really focused on one of those group of bryophytes, which are liverworts. So liverworts are amazing. Here, just a little display of how beautiful they are. They have so many morphologies. They live in many places. They live in many substrates. They're specialized in different substrates. Some of them are epiphytic. Some of them live only on rocks. Anyway, there's a lot of morphological diversity in this group. That accounts for about 5,000 species of liverworts worldwide. You can find them anywhere from like deserts, even in Antarctica, you can find liverworts. And from an evolutionary perspective, they diverge from other groups of plants more than 400 million years ago. So if we think about what that means, it means that since 400 million years ago, they have had their own trajectory, accumulating their own morphological characters, and have solved the same environmental conditions that vascular plants have solved in perhaps different ways. Something that I really like to think about is that not only they're super old, But if we look at the fossil record, for example, here we have Mercantites, which was found in the early Cretaceous. So that's about the time where angiosperms are starting to become dominant in terrestrial ecosystems. We found plants that are like super similar to some of the extant liverworts, which means for a long time, liverworts have kept the same morphology and have been successful using the same strategies they have used for millions and millions of years. So I'm going to argue that liverworts have a unique way of being a land plant. Some of these features of being unique are shared with the other two groups of bryophytes, namely mosses and hornworts. For example, they experience life as haploids, unlike vascular plants that experience life as diploids, so they have two copies of chromosomes. They disperse through spores, which is usually associated with having a high dispersal capacity because spores are smaller and get airborne. And they're poikilohydric. So that means that they adapt their internal water content to the environment, unlike vascular plants that try to keep their internal water content stable. Which means that they have to be desiccation tolerant when the environmental humidity goes down, right? So some people might think that cactus are desiccation tolerant, but they're really not. What a cactus does is they preserve their internal water. So the cells don't experience the lack of water because they store the water inside. Versus bryophytes, the cells fully dehydrate and survive those conditions and come back later on. Another characteristic is that liverworts and the other bryophytes tend to be small because they don't have lignin, but that doesn't mean they are not successful. In fact, they have been successful without having such important molecule like lignin in vascular plants. But even within bryophytes, liverworts have unique structures. So for example, they have ventral scales. We're going to come back to ventral scales later. They have these specific structures, unicellular structures, very small, that help disperse spores called elateres. They're only in liverworts. They have oil bodies, which are organelles. So there's an organelle that is only in liverworts. You don't find it in any other group of plants. These oil bodies tend to have a lot of molecules that we're only starting to study. All the time there are new molecules described that come from liverworts. Among them, there are like very interesting pigments like auronidins, which I'll talk a little bit about later that I'm very interested in. So they're a source of constant discovery as a lineage. All these unique ways of being a land plant that we can find in liverworts must have evolutionary implications, right? But when we think about the biology of plants, our understanding of what it means to be a plant and how plants evolve and what are the mechanisms of evolution in plants, most of our understanding comes from vascular plants and in particular from spermatophytes and angiosperms. Perhaps we need to start thinking a little bit more about what is different in bryophytes so we can actually understand the full extent of the variation of what it means to be a plant and how plants evolve. So my academic work involves trying to do a lot of evolutionary studies, focused particularly on liverworts to bring more attention to this type of organisms. So now I'm going to switch into talking a little bit about the biogeographic history of one family of liverworts, which is the first project I want to talk about. And I'm going to try to highlight the ways in which liverworts are different and why we need to use specific methods if we want to really account for the characteristics of a group like this. So as I mentioned, I work with this family Aytoniaceae, which is a worldwide family that has 6 different genera. As you can see, it's a complex fellow liverwort. It's extremely cute. And as a good liverwort, it disperses through spores. And therefore, we might expect that the distribution areas are very broad, which is true for some of those liverworts like Rebulia. But we also find other organisms that have smaller distribution ranges, and anything in between. Therefore, this variation makes this group really interesting to study how these different ranges evolve through time. Now, the way that we study geographical range evolution from an evolutionary perspective is using models like DEC. So basically, when you're doing an evolutionary model, what you do is you imagine how a particular thing changes through time. In the case of biogeographic models, that means we imagine what affects the range of a species. So we describe one type of change, which would be a dispersion. So imagine you have a lineage that occurs in the area A, and then after a dispersal event, now this lineage occurs in the area AB. Very easy, right? So there was a change, so therefore there was evolution. An opposite type of event would be an extinction, in which a lineage that occurs in the area AB, for example,
You finally got it.
gets extinct, and then goes into, now it's only in the area A, right? And something that is really interesting about biogeographic evolution is that we often think that it might be associated to cladogenetic events, meaning like at the moment in which a lineage splits is when the change occurs. So let's see with an example. We have a lineage that is in the area AB, and then at the moment of speciation, one of the daughter lineages keeps the area A and the other keeps the area B. This otherwise people might call it an allopatric speciation event, right? And we think these type of events are pretty common, so that's why these models try to account for them. So I wanted to use this type of model to study the evolution of the range of liverworts in the family Aytoniaceae. And we would do that. The way we work is like we take our species, right? We know where they are nowadays, right, in these areas. We apply the model, and we basically can reconstruct for the past nodes where these species could have been. Obviously, this is a simplification. It's a little bit more complicated than that, but that's basically how it works. Nevertheless, very early when I was trying to do this type of work, I realized that liverworts are special because they're very old, they're very widespread, and they have high dispersal capacities. All these characteristics make this model inadequate to study liverworts, and I'm going to explain you why. The first problem is that the DEC model cannot account for unobserved cladogenetic events. And I'm going to get a tiny bit technical, but I'm going to explain you with this example. So imagine we have a phylogeny, we have 3 tips, right? And 2 of them are in the blue area, and 1 of them is in the orange area. The way the DEC model worked is that it would try to infer how this happened. So it could explain this observation on the tips as an agenetic event, so something that happened along a branch. It could also explain the change as a cladogenetic event, so something that happened at the moment of the speciation that we are seeing here. But we know that when we reconstruct phylogenies, we often cannot account for all the cladogenetic events because some of the species go extinct, right? In that case, a DEC model wouldn't be able to account for this example, a change that occurred at the moment of speciation, but where one of the lineages went extinct, right? And we know that this problem becomes particularly bad when we're dealing with very old lineage, because as we deal with older lineages, there is more chances that this type of events will occur. So yeah, this was a big problem when I'm dealing with such an old lineage. Luckily, there is a framework, a different type of model that can solve this problem, which is called geodesy. Another problem is that given the implementation of the DEC model, we can only deal with lineages that have very small ranges, yet we know that liverworts tend to have widespread ranges. Luckily, there's another software that helps dealing with this problem. And if anyone is interested in the details of how this works, I'm happy to talk about it at any point. And the third big problem is that the DEC, as it's usually used, assumes that the rates are constant through time, which again is not feasible. It's not really what we expect if we have such an old lineage, right? But we can solve this by using a time heterogeneous. Anyway, there were multiple problems of using this type of model with liverworts, and there were individual solutions for each of the problems, but nobody had put together a model that combined all the solutions in a single implementation. So what I ended up doing during my PhD was getting super specialized and learning how to implement this type of models. So I developed a model that I called the Biogeography plus J. I'm not going to get into the details, but basically it's similar to DEC in that I define all the different events, some anagenetic, some associated with speciation, so some cladogenetic. And I'm not going to go into details at this point, but I just wanted to call your attention to the fact that my model distinguishes between areas that are close by and areas that are distant, right? And this is important for liverworts because, again, we think liverworts are able to do distant dispersal. The first step to do that is to get a time-calibrated phylogeny, which I did. And I just wanted to call your attention to the fact that this family of liverworts arose at some point in the early Cretaceous. So again, it's a very old group of plants. And we know that if we go back in time, the organisms have experienced different configurations of Earth, right? So we might expect that for all older lineages, the relationship between the areas matters, right? It's easier to disperse if areas are closer. It's more complicated to disperse if they're farther away. So my model also accounts for the relationships between the areas. So basically what I did is I had the continents and I went back in time and I defined the relationship between the continents, whether the areas were connected or were not connected. Anyway, implementing this model was a ton of fun. I had to code a lot of things. Again, I'm very interested in teaching this kind of models. Feel free to reach out to me if you want to learn more details. I'm always happy to talk about it. I implemented this model for my plants. It was a great achievement in my early career, and this model eventually ran. It took only 2 weeks, which is very fast for this type of analysis. And I'm going to present a few of the results. We're getting ready to publish this paper as part of a special issue in systematic biology. Anyway, so we learned a few details about the history of Aytoniaceae in particular. So we learned that there are some clades that are, for example, in Astrella, the older nodes were widespread, and then we have lineages distinguishing between North America and Europe. But some of the more interesting results, because we wouldn't be able to get this type of results with other model, is the fact that we infer a high probability of occurrence in Antarctica, for example. So from other evidence, for example, geological evidence, we know that Antarctica had a lot of diversity of plants in the past. This model, because it accounts for the movement of continents, infers a high probability of occurrence of liverworts in Antarctica, for example. But part of the cool things about these models is not only that we learn about the history of the specific group that we're interested in, but we also learn about the processes itself. So for example, if we want to know how common is for liverworts to disperse to adjacent areas versus to disperse to distant areas, we can put a number to that, right? So for example, we learned that liverworts in this family disperse to adjacent areas 10 times more often than they do to distant areas. So even for these good dispersers, distance matters, as we would expect. And on top of that, we're able to know the factor by which this event is more common, which in this case is 10 times. And these type of studies are even more interesting when we start thinking that we can apply to other groups, because this is for liverworts. What do you think it would be for ferns? What would it be for angiosperms, right? So that's when we get into very interesting questions. Another interesting result is that if we compare the type of dispersal, so imagine there's a dispersal to one area. What is the probability that that dispersal is associated with a speciation event, depending on whether it's adjacent or distant? We found that when the dispersal is to a distant area, there is 5 times more chances that it's going to end up in a split of lineages than when it is to an adjacent area. This makes all the sense, biologically speaking, since as you disperse to distant areas, there's more chances that the lineages are going to be isolated and therefore they're going to become independent. And again, the cool part is that we're able to put a number and know the factor by which these events are different. Again, it would be really interesting to apply this model to other groups. I adapted it particularly to the biology of liverworts, but there are other groups that share characteristics with liverworts of being very old, widespread, and having good dispersal abilities. So anyway, this is a project that I worked during my PhD, and this is the type of models that I like. And yeah, hit me with any questions regarding this type of models. Now I want to transition into the second project that I wanted to present you. So now I'll show you how understanding the biology of liverworts helps us to define and use methods adequate to the group. And now I wanted to explore that at a smaller scale. So let's zoom in into one species of liverworts in the same family. This project was possible thanks to the California Conservation Genomics Project, which is a statewide project that gave us funding for doing this research. So Calisterella californica is a complex sallow liverwort. Here you can see you have male plants, you have female plants. It's a dioicous species in the same family I studied previously. It is all over California, so there's a good chance that if you're hiking around, you have seen it. So the fact that it's all across California means that it's equally happy in wet environments like the redwood forest, but also in the desert. Like I found it. That picture is a picture I took in the place where I collected this plant. So that is pretty surprising, right? It means that this liverwort is actually very versatile and it's desiccation tolerant. In fact, I wonder how many of you have seen the plants like this in the field, right? Sometimes we see that and we don't know what it is. It is the liverwort that is desiccated and it's rolled over. But it's alive. If you put a little bit of water on one of those stelos, it's going to quickly open and become green again. Another super interesting fact about this species in particular is that it diverged from its closest relative 98 million years ago. So that's about the time in which the redwoods separated from their sister groups. That's about the time in which the eudicots separated from the other angiosperms. So it's a very, very old lineage, and Calisterella californica is the only representative of that lineage and is endemic of the California touristic province. So it's a pretty special species. So given that this species is that old and we have only described one species for that lineage, I was interested in knowing how diverse genetically this species is. Perhaps the underlying question is it really just one species? And we're going to get to that. So for this study, I collected Calisterella californica across its range. I got a total of 110 samples, for which I made voucher specimens and permanent slides. These are the points where I collected it, and it was really important, and I put a lot of effort in making sure that the collections spanned the geographic and environmental range of conditions in which this species occurs. I did a full genome resequencing of all these 110 individuals, again, thanks to CCGP. And I wanted to mention that when we're working with non-model organisms like most liverworts, there's always extra work that we need to do. So in this case, we had to do a reference genome to be able to process the information from the other samples. And this is really cool because now this species from California is the fifth liverwort that has a nuclear genome, which opens the door to a bunch of studies. And this is a great opportunity to show you a baby liverwort. I grew these liverworts from spores, and this is how a baby liverwort looks. And look at the scales. So even since they're super tiny, they already have ventral scales. We're going to get back to these a little bit later. So coming back to the question, I wanted to know how much genetic variation it is within this population. For that, I used that SNP approach, basically looking at differences in genetic markers. And I use a admixture analysis. So basically we have here a list of all the samples. And what this analysis does is that it infers the ancestry of each of those individuals. And here they're color coded. So you have some individuals that are like one single color. Some of them have mixed ancestries, right? This doesn't make a lot of sense there, but when we put it in a map, we start seeing patterns, right? So we see a geographical differentiation. So we have in the coastal region, mainly the purple individuals. In the Sierras, we have the blue individuals. And then in Southern California is where we find these mixed individuals, this green population. And I don't know if it's very clear from the map, but these yellow populations, these yellow individuals are the desert ones. When we put these individuals in a principal component analysis, which arranges by similitude, by how similar the individuals are, we distinguish the desert individuals clustered together, the Sierras individuals clustered together, and all the rest are together. So basically, the coastal and the southern individuals are clustered together. So at least we're having three groups very differentiated. I then applied another analysis, which is a little bit of a more sophisticated version of a mixture that accounts for the geographic location of each individual. And we find very similar results. So again, a coastal population, a Sierras population, and a southern population. So at this point, I am pretty convinced that we have an important differentiation, genetically speaking, of three groups of these liverworts. But the question comes to, does this genetic differentiation requires describing new taxonomic groups? In this project, I still need to do more work. So I still need to do some gene trees. I still need to do morphology. I have looked at vegetative characters and there's no differences in the thallus. So now I'm looking at spores. And in preparation for the possibility that we might need to describe new species, I have worked with some collaborators and we have revised the typification of these species. So we know where the name should go in case we describe new variation, new taxonomic groups. And this is going to be continued, but a little teaser, because I started to do some SEM imaging of the spores of these plants. So look here, these are spores from the coastal population in the top row and from the Sierras population in the bottom row, and the difference are quite striking, right? So obviously I need to do more individuals. And something important to mention is that we don't have spores from the desert. The plants in the desert very rarely produce spores and they're not really collected. So if you happen to be doing floristics and you happen to collect Colastrella californica in the desert area and you find spores, please, please, please let me know, because that would be a very good complement to this work. Okay, so moving into the last part of this talk, I have shown you the type of studies that I do with liverworts, how I want to study at different scales. And I'm gonna now move into what I'm starting to do, because these two other projects are close to be done. So as I mentioned earlier, when I was collecting Calastrella californica, I saw it in two versions, right? The wet, happy version, the desiccated version, that although it looks sad, is still alive. What happens when you see the desiccated version is that you're basically seeing the scales of the liverwort. It rolls over. And perhaps it's easier to see if we look at this video. So here what you're seeing is you're seeing the liverwort rolled over. So you're looking at the scales. And once you hydrate it, this is how it looks like, right? So one more time, it rolls out, and now you can see the liverwort. So basically, the whole desiccated time, which is in California all summer and all winter, the liverwort is just inside its ventral scales. So after seeing that, I started thinking and getting super interested in ventral scales. So a little bit of background about ventral scales. We rarely see them, and that's because when you see a liverwort, you often see the top part. And to see the scales, you would have to flip the liverwort, and you would have to clean all the dirt from underneath, and then you would see the scales. The scales are usually arranged in rows, so often 2 or 4 rows, and we don't know much about them. So something that has been said in the literature is that scales help the attachment of the plants. But I believe there are more functions. So for example, one of the functions is, I think in some liverworts, they might help to the external conduction of water. You can see these very large scales in these Mercantia veteroana. And they can also have protective functions. So I already told you about the rolling over in Calastrella californica, but in other species, the ventral scales protrude from the tip of liverworts, and this is exactly where the meristematic cell is, right? So I can totally imagine that these scales are protecting the meristematic cells of liverworts. And then, so I started getting super interested about scales. So I started like looking how the scales look like in other species beyond Calastrella californica. And here, just a representation of scales variation, right? I have arranged them in like 2 axes. So they vary in size, for example. This is almost at a correct scale. So they are like very small, they can be large, but they also vary in pigmentation, which we're going to discuss in a sec. It's important. Other axes of variation are, for example, the number of appendices, the shape of the appendices, which vary from like linear to these rounded appendices, the number of ocelli. So there are many axes of variation. If we want to kind of make an exercise, a thought exercise, and condense all the variation in one axis, I will argue that we will find in one extreme scales like these, which are super small, translucent, so not pigmented at all, and without appendices. And in the other extreme, we would find scales that are comparatively much larger, with a lot of appendices and super pigmented. And even more interesting is that the liverworts that tend to have these small scales tend to be in very, very wet environments, whereas the liverworts that tend to have this other extreme of scales tend to be in very dry environments. So I really think that scales in liverworts have very important functions, from protection to water conduction to attachment, and that the different functions that these scales perform help them adapt to different type of environments. So I think scales have not only a role in the physiology of organisms and the survival, but also in the distribution range of organisms, right? And I think this is a good time for me to give like a little shout to my students, Ria and Atra, who published their research in our society journal, Madroño, They worked studying the scales of liverworts. So they contrasted a xerophytic liverwort with red scales versus a neutrophilic liverwort with translucent scales. And they found that the red scales of Calastrella californica protect against UV radiation, or at least have the potential to protect against UV radiation. So again, this is another axis in which the scales may be having a very important role in the liverwort life cycle. And not only they published the research, they actually won a prize in the last student symposium of the California Botanical Society. So I'm very proud of them. You can read their paper there. So anyway, coming back to scales and the diversity of scales, again, I'm an evolutionary biologist, so I like to look at scales in a phylogenetic context. So I wanted to see how these traits are distributed in a phylogeny, just a quick exercise. So we can see a few things. We can see that the organisms that have superpigmented scales tend also to have very big scales and tend also to have the rolling mechanism. The other good news for an evolutionary biologist is that these traits are spread in the phylogeny, which means this is a good system to apply the kind of models that I like to use to better understand the evolution of this group. Therefore, the project I'm starting to develop, that is going to be my main research in the next few years, is to first document the morphological and functional diversity of these ventral scales in liverworts. Then I want to understand the evolutionary patterns of the scales in these groups and the implications for the geographic range of liverworts. As you heard at the beginning, I love models. So this project is going to involve developing a new model that allows us to study at the same time the evolution of geographical range and morphological characters, so we can understand how they interact and answer questions of the type, do purple scales help liverworts to live in desert environments from an evolutionary perspective? So that's the type of questions I'm interested to work in the future. I'm going to do this in RefBase, which is the platform I'll be using. And I hope in this talk you learn about this amazing group of plants. Many of you probably know a lot about them. I really think it's important that we bring them into the evolutionary, but also conservation focus. And with that, I just want to thank to all the people that have worked with me in many of these projects, all my funding sources, and thank you for being here today. And I'm very happy to take any questions. We'll stop sharing.
Thank you so much for a wonderful talk. That was super interesting. And I kind of also wanted to point out that your undergrads won the award at the Kalbat-Sock Symposium recently for that work that they presented a poster on the work that you mentioned. So it was fun to see you present that as well. I think that there are a bunch of questions, so we can start with Peter Alpert.
Very nice, Ixchel. I was wondering about the possible role of substratum in the evolution and distribution of the liverwort. So, for example, are the ones that occur on plants, do they have differences in the range compared to ones that occur on rock? And do you find a lot of substratum switching? How does it map onto phylogeny?
Yeah, so I think a lot of these questions are really hard to address, first because we don't know a lot of those details. When you were saying about substratum, though, the one thing I thought is the ones that are epiphytic are usually leafy liverworts. And we already know that leafy liverworts tend to be much more diverse in the tropics. So that would be like, at least biogeographically speaking, you would find all these leafy, all these epiphytic specialized clade being very abundant in the tropics, but not in the higher latitudes. In terms of the substrate, at least what I have observed with the complex phthaloids liverworts, which is the group that I work with the most, is they don't seem to care much about substrate. It's more the conditions of how exposed is the rock, how much sun they get. The type of rock itself doesn't seem to matter a lot about, at least for complex phthaloid liverworts. And I'm not sure if I am missing one of. There were a few questions, Peter. So let me know if I missed.
So they all pretty much occur both on rock and on soil.
Yeah, at least for the complex phthaloids that I study, yes.
Thanks.
Sure, thank you.
Andy Simpson?
Video is still locked. How confident? So I had two questions. How confident are you about the age of the separation of Calistrea from its sister clade? You had 98 MA, but what are the error bars on that?
Oh, I can go back and tell you right now. I'm pretty confident that it's probably. Let me actually share it. Oh, I don't have it here. I am pretty confident that it's on that range. 1 sec. I have one with error bars. Did I? Oh yeah, there we go. Some share. There we go. This one is it's with the error bars, right? So I had 98 on the other one.
So it's Cretaceous, but we're
still
not sure
Cretaceous,
when
right?
in the Cretaceous. Okay.
Yeah, so it could be a little bit, well, it could vary in all these. Obviously, we don't know the shape of this. I think it's probably something like this, so it's more likely around this range. There's variation, but it's still pretty old. So it's still up in the ballpark of like the split of Sequoia. So it's still a very different, like a very deeply divergent lineage. Yeah.
Well, this relates to the other question that I wanted to ask then, which is how long ago do you estimate the divergences between the different genetic versions of Calistrea californica? And the reason why I ask this is because California, the landmass that is California, was mostly assembled over the Cretaceous and into the early Cenozoic. So
Yeah.
I'm wondering if those divisions are themselves ancient, or if that's a much more recent spread.
That is a great question, and that is a question that I do want to address, but I don't have the data for it yet. Because what I have to do is to get trees, right? Like right now, I just have genetic differentiation, but what I need is really make a phylogeny and date that phylogeny, and I'm doing it. It just takes time to extract the markers, but as we speak, it's running. So I'm eventually going to get genes and I'm going to be able to address that question of how old they are, because it's super interesting, right? Like it could be that it was an ancestral population and then it split, maybe from the south to the north or whatever. Or it could be that they were like ancestrally differentiated and just convergent recently.
If it developed geologically as the state developed, I would expect the Sierra Nevada populations to be the oldest, because that's the part of California that already existed by the Cretaceous.
Yeah, the other thing is like we don't know anything about where exactly they came from, right? Like now they're in California, but were they in California in the past? So that would require like a larger study.
Much of the modern day California flora appears to have originated in Nevada during the Miocene. So that's another possibility, is if it's a much more recent invasion, it came from the east.
Yeah, and it's really interesting that there's a lot of plants that seem to have this pattern of genetic diversity. Like I know at least I think Was it iris? No. It was one lily that also had the same pattern of like very differentiated populations in the Sierras and in the coastal region. Yeah, thank
Adrian?
you for the question.
Yeah, I had a
Oh
couple
yeah,
of
californicas.
questions. Really cool work. I was curious if you saw a significant difference in ventral scale and aranidin pigmentation between male and female.
No. So far, no. We haven't seen differentiation in ventral scales. Because I went through the pain of not the pain, but like cleaning scales for every single collection I made. So I have slides for every scale. And when I was cleaning, I didn't see any differences, evident differences, but I have the material to go back and look at the morphology. It would be really interesting to see if the morphology of scales matches either the genetic variation or sexual dimorphism as you are suggesting. But I think that's going to have to be like a work for a student, a student that wants to work on ventral scales in Australia, californica. I have the materials ready to go. But yeah, so I didn't see any on the first pass.
And that's also for the same case for the aranidins. They would have the same pigmentation depth.
The Arinodines. I don't think we can measure Arinodines just from looking at the scales. Or I guess
I'm asking if when you actually measured the aranidins, was there a difference between the male and the female accession of the same species?
We didn't look at female versus male. We just pulled multiple plants in that particular experiment. We just pulled multiple plants. So no, we didn't look at sexual dimorphism. So I don't think I can answer that question. Yeah.
And then my second question is, my understanding aranidins are released from like carbon-nitrogen imbalance. And I was curious if you had tried growing these liverworts in the same environments and seeing if they release the same pigment. I guess I'm trying to get at if the aranidins are sort of like a fingerprint to the species, that there's a unique hue to each individual species.
Oh, that's super interesting. I have not even thought about that. As far as I have observed, just from like looking at specimens, the pigmentation it's a species trait, but within the same species, there's a little variation. So even in Calastrella californica, another species that would have them is Darjonia, for example, you can see probably different concentrations, because the coloration varies, right? Like very exposed individuals tend to be more purple, tend to have purpler scales than less exposed individuals. But when you say
exposed, what do you mean?
To sun, for example, like if they're in a very dry environment, exposed to a lot of UV radiation, they tend to have more pigmentation than the same species in a more mesic environment. But the variation is larger within species, right? So it's definitively a phylogenetic component. So it's definitively species dependent, but within the same species, there's a little variation. And I did have no idea about that nitrogen carbon imbalance. So if you have any reference, I would be curious.
Yeah, I'm also a liverwort biologist, so I can forward you a paper.
Yeah, that'd be super cool. The other cool thing that I didn't mention in the talk, but I read a paper about it recently, is that the pigmentation of the oneridines tend to deposit in the cell walls, and that makes cell walls more resistant. So the coloration the pigmentation not only protects, again, UV radiation, but it also gives structural sturdiness to the ventral scales, which is super cool.
Yeah, from my understanding, it's like a carbon sink where it just channels its resources. and just packs it into the cell wall.
Okay, that's super cool. Thank you.
I have sort of a related question from a non-liverwort biologist about the pigments. I was curious if they're similar to the anthocyanins that other vascular plants are using to make purples, or are they a different class of pigment? Or
Different class.
Yeah. My understanding is that they're anthocyanin class, but they're their own thing, and they have only been found in liverworts, as far as I know. And it's not very long ago that they described them, so that's what I say. Like people keep finding new molecules in liverworts all the time.
That's super interesting.
Thank you.
Other questions? If no one else, I have another question about your model, your biogeographic model.
Sure.
I was wondering, is that a flexible model that you can adapt to groups that have different biogeographic regions and you just kind of need to know their arrangement through history? Or is this something that other people can, I guess more broadly, is this something other people can use to adapt to their system?
Yeah, so basically what you would need to know is you would need to define the areas that obviously are organism dependent, and you would need to define the connectivity of areas through time. That is, but yeah, you could adapt to your group. Yeah.
That's awesome.
Yeah. Thank you.
That up. Very useful. All right. Well, if there's no other questions, then let's give Ishel a virtual round of applause. Thank you so much for giving this talk, and it's nice to see everybody. And next month we have Carrie Tribble on board for giving the monthly seminar. So be sure to check back in on, I believe that's July 10th.
Thank you so much.
Yeah, thank you.
I have a last minute personal question.